Novel biomaterial design for targeted arthritis therapy. Researchers have developed a novel hydrogel material that holds significant promise for revolutionizing the treatment of chronic conditions, particularly arthritis. This engineered material possesses the unique ability to sense minute changes within the body, such as those that occur during an inflammatory episode, and subsequently release therapeutic drugs precisely where and when they are required. The key to this mechanism lies in the material's responsiveness to subtle shifts in pH levels. During an arthritis flare-up, the affected joint becomes inflamed, leading to a slight increase in acidity, a lower pH, compared to the surrounding healthy tissue. This acidic change acts as the biological trigger for the drug delivery system, allowing for highly targeted intervention. The development of such a smart material represents a major step forward in creating more efficient and less invasive treatment options for millions of people worldwide living with debilitating joint pain and inflammation. The material developed by a team at the University of Cambridge is essentially a squishy, jelly-like polymer network capable of encapsulating anti-inflammatory drug molecules. Its design is specifically tuned to react to the narrow pH range characteristic of an inflamed joint. As the acidity increases, pH decreases. The material undergoes a predictable physical transformation. It becomes softer and more compliant. This change in its mechanical properties directly initiates the release of the drug molecules held within its internal structure. By confining the release of the drug to areas with this specific acidic signature, the researchers aim to achieve highly localized drug delivery. This precision is expected to be a major advantage, as it could substantially reduce the potential for systemic side effects that often accompany conventional drug administration, thereby improving the safety profile of the treatment. The primary envisioned application for this smart material is its use as an artificial cartilage within arthritic joints. If successfully implemented, this approach could enable the continuous and automatic treatment of arthritis. Instead of relying on periodic oral medications or injections, the artificial cartilage would serve as an on-demand reservoir, releasing medication only when the inflammation process signals a need. This method promises to significantly improve the efficacy of drugs in reducing pain and combating chronic inflammation, two primary goals of arthritis management. The public health burden of arthritis is substantial. For example, it affects over 10 million people in the UK and globally is estimated to impact more than 600 million individuals. Improving treatment outcomes for such a vast population could lead to major benefits for healthcare systems and patient quality of life. The research team's findings, which detail the fundamental principles and initial testing of the material, were published in the respected Journal of the American Chemical Society. The hydrogel's sophisticated behavior is rooted in the use of specially engineered reversible crosslinks within its polymer network. These crosslinks are the structural components that give the material its mechanical integrity and sensitivity. The sensitivity of these links to changes in acidity levels is the fundamental scientific principle that grants the material its remarkable responsiveness. The development of this technology took place in Professor Oren Sherman's research group in Cambridge, which specializes in designing and building unique functional materials for diverse applications. Professor Sherman, a specialist in supramolecular and polymer chemistry, noted the long-standing interest his group has had in using such materials in joints because their mechanical properties closely mimic natural cartilage. Combining this structural function with highly targeted drug delivery is considered a significant and exciting scientific breakthrough. The most compelling feature of this system is its autonomous operation. As first author Dr. Stephen O'Neill highlighted, these materials can effectively sense when a disease state, like inflammation, is active and respond by delivering the necessary treatment directly to the affected site. Crucially, unlike many other proposed drug delivery systems which need external energy sources such as heat or light to trigger drug release, this hydrogel is powered entirely by the body's own chemistry, the inflammatory pH change. This inherent self-regulating mechanism is expected to reduce the need for repeated doses of drugs while also enhancing the overall quality of life for patients. The researchers believe this novel method could pave the way for longer-lasting targeted arthritis treatments that automatically react to flare-ups. thereby boosting effectiveness while minimizing the potential for harmful side effects throughout the rest of the body. Initial laboratory tests have demonstrated the material's intended function. Researchers encapsulated a fluorescent dye within the hydrogel, which served as a model for a therapeutic drug, and monitored its release under different conditions. The results confirmed that at acidity levels typical of an arthritic joint, the material released a substantially greater amount of the encapsulated cargo compared to conditions simulating normal, healthy tissue pH. Co-author Dr. Jade McCune explained that by carefully tuning the chemistry of these gels, they can be made highly sensitive to the subtle, specific shifts in acidity that define inflamed tissue. Furthermore, the researchers suggest the approach is highly flexible and can be tailored to a wide range of medical conditions beyond arthritis, including certain types of cancer. This flexibility means that the material could potentially incorporate both fast-acting and slow-acting drugs, offering a single treatment device that could last for extended periods, from days to weeks or even months. The immediate next phase of this research involves testing the materials in living systems to thoroughly evaluate their performance, biocompatibility, and safety within a real physiological environment. While extensive clinical trials are required before the material can be translated into patient care, the researchers are optimistic that their approach could significantly improve treatment outcomes for people suffering from arthritis and other chronic diseases. The research was supported by key funding bodies, including the European Research Council and the Engineering and Physical Sciences Research Council, EPSRC. If the subsequent testing proves successful, the team believes this technology could ultimately open the door to a new generation of responsive biomaterials capable of treating chronic diseases with unprecedented precision and improving patient management globally.